Welcome back to my channel Knowledge Chronicle.
Today, we're diving into the fascinating world of genetics, exploring how traits are passed down through generations. We'll unravel the mysteries of Mendel's experiments, understand Punnett Squares, and delve into concepts like incomplete dominance and codominance. Let's embark on this genetic journey together. Gregor Mendel, the father of genetics, conducted groundbreaking experiments with pea plants. He crossed a true-breeding tall plant TT with a true-breeding dwarf plant TT. The first generation F1 offspring were all tall TT, showcasing the dominant trait. This experiment laid the foundation for understanding how a single gene influences a trait. Genotype refers to the genetic makeup TT, or TT, while phenotype is the observable trait tall or dwarf. In the F1 generation, all plants were tall phenotype with a TT genotype indicating the dominance of the T allele. When F1 plants TT were self-pollinated, the F2 generation displayed a 3-to-1 ratio, 3 tall plants to 1 dwarf. The genotypic ratio was 1 TT to 2 TT to 1 TT. This demonstrated that the recessive T allele was not lost, but masked in the F1 generation. Dominant alleles mask the effect of recessive ones. In our example, T is dominant over T. Thus, TT individuals appear tall, even though they carry the dwarf allele. Mendel's law of segregation states that allele pairs separate during gamete formation and randomly unite during fertilization. This explains the 3-to-1 phenotypic ratio observed in the F2 generation. Developed by Reginald Punnett, this tool helps predict the probability of offspring genotypes. By placing parental alleles on the axes, we can visualize all possible genetic combinations. To determine if a tall plant is homozygous TT or heterozygous TT, it's crossed with a dwarf plant TT. If all offspring are tall, the parent is TT. If there's a mix, it's TT. This is known as a test cross. In some cases, neither allele is completely dominant. For instance, crossing red RR and white RR snapdragons results in pink RR offspring. This phenomenon is called incomplete dominance, where the heterozygous phenotype is a blend of both parents. Codominance occurs when both alleles are equally expressed. A prime example is the ABO blood group system. Individuals with genotype 1AIB express both A and B antigens, resulting in AB blood type. Some genes have more than 2 alleles. The ABO blood group has 3 IA, IB, and I. The combination of these alleles determines an individual's blood type, showcasing the complexity of genetic inheritance. A single gene can influence multiple traits, a concept known as pleiotropy. For example, the gene responsible for sickle cell anemia affects red blood cell shape and can lead to various health complications. Genetics is a captivating field that explains the diversity of life. Understanding these principles helps us appreciate the intricate mechanisms of inheritance. If you found this video insightful, your one like, share, and subscribe will help us create more valuable content. Thanks for watching. Stay curious.